on the basis of morphology of mycelium, mode of spore formation and fruiting body, the members of kingdom fungi are divided into Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. The first group of fungi, the Phycomycetes, have cenocytic and aseptate hyphae. Aseptate means without septa or cross walls, while the other groups have branched and septate hyphae. In Phycomycetes, sexual spores are called zygospores. In Ascomycetes, they are called ascospores. The members of Basidiomycetes form basidiospores, while in Deuteromycetes, sexual spores are absent. Regarding the fruiting bodies, in Phycomycetes, there is no complex fruiting body, but the zygospores are formed in zygosporangium. In Ascomycetes, the fruiting body is an ascocarp, which is cup-shaped. In Basidiomycetes, the fruiting body is a basidiocarp, while in Deuteromycetes, the fruiting body is absent. Let's see the first group in kingdom fungi, the Phycomycetes. They are found in aquatic habitats, decaying wood, or as parasites. For example, the white rust on mustard leaf is due to Albugo candida. The mycelium of the members of Phycomycetes is cenocytic and aseptate. The word aseptate means without septa or cross walls. I hope you remember this picture of the multinucleate hyphae. Asexual reproduction in Phycomycetes involves production of asexual spores such as zoospores and aplanospores. Zoospores are motile, while aplanospores are non-motile. Spores are formed endogenously, meaning produced inside a structure called sporangium. When the spores mature, the sporangium breaks open and releases all spores. And each spore will germinate into an individual fungal hypha. About the sexual reproduction in Phycomycetes, two genetically different hyphae will come together. At the tips of these hyphae, gametangia bearing gametes will be produced. One hypha will produce a male gametangium with male gametes, and the other hypha will produce a female gametangium with female gametes. The male and female gametes fuse to form the diploid zygote. The fungi is said to be isogamous if the fusing male and female gametes are of the same shape and size. Here, both the gametes are motile with flagella. In the case of anisogamous, one gamete is very large in size when compared to the other. Here also, both gametes have flagella and are motile. In oogamous type of fungi, one gamete is very large in size when compared to the other. That sounds very similar to that of anisogamous fungi, but the difference here is that only one gamete, the male gamete, is motile. The large female gamete is non-motile. After the formation of zygote, it transforms into a large zygospore. The zygospore may stay dormant for long periods of time, but when the conditions are favorable for fungal growth, the diploid nuclei undergo meiosis to make haploid spores. These spores will germinate into a haploid hypha. And can you tell me if this hypha will be genetically similar or different to their parents? The answer is yes, it will be genetically different because it was a result of sexual reproduction. Now let's move on to the second group in kingdom fungi, the Ascomycetes. They are called sac fungi because their sexual spores called ascospores are produced in a sac-like structure called ascus. Ascomycetes can be unicellular like yeast or multicellular like penicillium. Some of the other members of this group belong to the genus Aspergillus, Claviceps and Neurospora. Neurospora crassa is used as a model organism for numerous genetic, biochemical and molecular studies. This is possible because the genes of Neurospora crassa are all sequenced. Also, this fungus is very easy to grow and has a haploid life cycle which makes genetic analysis simple. Some other members of Ascomycetes like morels and truffles are considered to be expensive foods. They are expensive because they are found in the wild and it is not easy to cultivate them commercially. About truffles, these fruiting bodies grow underground and trained dogs are used to find them using their characteristic smell. The truffles are then dug out of the soil. According to the nutritional modes of Ascomycetes, they can be divided into saprophytes that feed on dead organic matter, decomposers that break down complex organic matter into simpler forms, parasitic fungi that depend on living organisms, and another group, coprophilous fungi. The word coprophilous means dung-loving. These fungi live in animal dungs. Sounds strange. The secret here is animal dungs provide an environment rich in nitrogenous material and there is minimum competition from other organisms. I think they are brilliant. The mycelium of the Ascomycetes fungi is branched and septate, meaning with septa or crosswalls in the hyphae. The asexual spores are conidia, which are produced on conidiophores. The sexual spores in Ascomycetes are called ascospores and they are produced in sac-like asci. Asci is the plural form and ascus is the singular form. The asci are arranged in the cup-shaped fruiting body called ascocarp. A full view as well as a longitudinal section of the ascocarp is shown here. Let's move on to the third group of kingdom fungi, the Basidiomycetes. Commonly known form of Basidiomycetes are mushrooms, bracket fungi and puffballs. They live in soil, decaying trees or in living plant bodies as parasites, for example rust and smuts. Some are edible and some are deadly toxic. Amanita species, which are one among the most toxic fungi in the world, belong to Basidiomycetes. The mycelium of Basidiomycetes is branched and septate. Asexual spores are not generally found in Basidiomycetes. Other than sexual reproduction, they show vegetative reproduction by fragmentation of hyphae. In the sexual reproduction of Basidiomycetes, basidiospores are produced in spore-bearing structures called basidia. The singular form of basidia is basidium. Basidia are arranged in the gills of the basidiocarps, the fruiting bodies. Basidia are club-shaped. Definition for a club shape is narrow at the base and broader at the tip. Because of the presence of club-shaped basidia, members of Basidiomycetes are called club fungi. Moving on to the last group, Deuteromycetes. They are called imperfect fungi because only asexual and vegetative phase is known for these fungi. If sexual stages are discovered, then they are moved into Ascomycetes and Basidiomycetes. They produce asexual spores called conidia. Their mycelium is septate and branched. Based on the nutritional modes, Deuteromycetes can be divided into saprophytes, parasites and decomposers of litter helping in minerals recycling. Some members of Deuteromycetes are Alternaria, Coleotrichum and Trichoderma.